Hello, and today I'll be presenting the paper Bespoke Plant Glycoconjugates for Gut Microbiota Mediated Drug Targeting. This paper was written by Wei-Zhen Ma et al., published in Science Magazine, May 1st, 2025. This is a study done on trying to find a better drug targeting method for people suffering from Crohn's, IBD, and other large intestinal diseases. Why is this important? The bottom line is this therapy essentially utilizes the microbiota in our gut to be more effective in assisting with medicine delivery. This therapy was developed mainly to treat IBS, IBD, and Crohn's disease, but the applications can be utilized for other gut-mediated diseases, like the researchers state in the article, like colorectal cancer. Some background, previous studies done. A study conducted in 2021 by Briggs et al. Showed that human microbiota can utilize glycan, which bypasses the human digestion tract since we cannot process it. We don't have the genes to break down glycan, so the methodology used in this paper was to kind of hack the way the system naturally behaves so that we could bypass the primary digestive areas. And then a study from 2014, which this paper leaned on, showed that the bacteria species Bacteroides can process complex plant polysaccharides, which is how we get the nutrients from those specific plant products when we eat plants and plant-based polysaccharides in our diets. Some background on what Crohn's disease is. Crohn's disease is a bowel disease in which there is a constant inflammation and irritation of the intestinal lining. This is usually characterized by abdominal pain, diarrhea, and weight loss. It is a very chronic condition with no known cure, and treatment has been developed to help with symptoms and improve QOL, which is quality of life. But obviously, like I said before, there is a chronic condition with no cure, so the symptoms can be abated, but typically it's with a lot of medication that, while it improves the quality of life, is very debilitating to the people who are taking it. On here, I've provided a graphic that shows what Crohn's disease looks like in large intestine and what ulcerative colitis looks like. With ulcerative colitis, you have the back end of the large intestine being affected. With Crohn's disease, you have various inflammatory spots spread out throughout the large intestine, which leads to chronic issues and trying to deal with that passing. Ulcerative colitis is just as bad in terms of like where it goes, and this drug development that they came up with could treat both of these conditions. What is a glycoconjugate? A glycoconjugate is a complex plant oligosaccharide. The Bacteroides species encode a gene that targets the glycoconjugates utilized in this study. Here I've provided part of the graphic that will be shown later, and I'll go over it at that time. What is glycocaging? The team in the paper dubbed the process they developed glycocaging. Glycocaging refers to the process of caging an anti-inflammatory drug, commonly used to treat IBS, IBD, or Crohn's, to bypass the stomach and be delivered to the microbiotic elements in the lower intestinal tract or the large intestine to release the drugs where it can be more effective. So essentially what it does is it adds the oligosaccharide to the IBD drug and allows it to bypass the normal portion of digestion. So normally these drugs would be digested and then distributed through the blood system. This allows it to be bypassed into the gut directly, into the large intestine directly, rather. The necessity for this. The way most more oral medications work currently, the stomach and small intestine break down the majority of the drug, and most of it gets absorbed through them. It then gets distributed through the bloodstream to where it needs to go in the locations it needs to go to. But this leads to anti-inflammatories not being able to reach the large intestine most of the time where the drug is needed, because by the time it gets distributed to the large intestine through the blood, it's been diluted, processed by other parts of the body, and generally not as much of an impact. The study gave us a method of getting the drug where it needed to go successfully by bypassing the stomach and small intestine using glycocaging. It also was found to be successful at lowering the amount of medication necessary, thereby reducing the amount the body has to attempt to break down to absorb. So this would also improve the amount of medication that we could use by making it so that we don't have to use as much when we go to take those medications or those drugs. So it helped reduce the medication amount necessary to induce a response in the subjects being tested. So, quick article breakdown. The team first had to identify what type of glycoconjugate could be broken down by the Bacteroides species, and after their own research, they discovered that zygoglucans, which are highly branched polysaccharides produced by cell walls of fruits and vegetables, are digestible to the Bacteroides species. So, on the right hand side of the slide here, we have zygoglucan, which is that yellow stripe wrapping around the cell wall structure of the plant cells. And then upon further investigation, they found the targeted gene in Bacteroides, the zygoloci, which is a multi-gene trait. That is pictured on the left here. Basically, it is the genes on the left-hand side of the figure that process and digest and break down the stuff on the right-hand side of the picture, which we'll talk about when we get to the results page, because this is one of the figures in the results. They also ensured that humans did not carry the zygote gene prior to moving forward. Obviously, if we're trying to bypass the stomach and small intestine, we have to make sure that humans can't actually break this down and make sure that we don't carry the gene that would allow us to do so. This ensured that the delivery method for the drugs would not be hampered by the human gut interference. So, obviously, like I said, here's a picture of the digestive tract. Oral medication goes into the mouth, you swallow it, goes down the esophagus and into the stomach. This is where the first or the second part of digestion happens. Obviously, the first part is the saliva. But then here in the stomach, it gets broken down. Small intestine breaks it down further. And large intestine, completes the breakdown and grabs whatever nutrients are left before it exits the system. This delivery method ensured the drug bypassed the stomach and the small intestine by using the zygoglucan, to get it to the large intestine. Methodology. First, the team had to develop a logical and usable glycoconjugate. They tried multiple different cages, but eventually settled on the XXXG glycoconjugates in their studies, specifically XXXG, which they combined with dexamethasone, budesonide, and prednisolone in order to test those specific drugs. So on here we have the structure of the zygoglucan and the beta sheet omega glycoside. They tested their developed drugs in a liquid medium with live Bacteroides cells in vitro, and they kept the range within 0.1 to 2 grams per liter of concentrated XXXG dex, bud, or pred, to show that all concentrations they were going to test would dissolve in vitro with beta cell or with Bacteroides cells. They also decided to test a 5 gram per liter solution and observed a time-dependent release, showing that the release method should translate from in vitro to in vivo. So basically what they ended up doing with this part is they tested all their concentrations they were going to test in the mice models to make sure that it would happen with the Bacteroides cells in an in vitro environment. When they saw the success there, they knew they could move forward. And then they also tested the maximum dose of what people get prescribed regularly, and showed that it would be a time release, meaning it would release the medication on a time release basis. So we went there. They went there and made sure that it would work with just bacterial cells, and then also that would translate actively into living cells. All right, so now we're going to start breaking down the results. So as you can see in figure 1, we have the first part of the figure, which shows the breakdown of the zygoglucans by the Bacteroides species. It attaches to a protein on the exterior membrane. It's transported across the membrane, broken down by various proteins and enzymes inside of the Bacteroides cells, and then releases the byproducts on the other side through a different pore. And then in section B, here we have the labeling of the gene specifically that breaks down the zygote in each of these different Bacteroides species. They tested multiple different Bacteroides species to make sure that the results would be bordered across the would be even across the board in regards to which species would be able to target the zygoglucan and allow it to um, process the medication efficiently. Still figure one, but we're going to break down C, D, and E now. C shows you the chemical structure of the triple XG type glucan, zygoglucan that was utilized, and then also the triple XG beta sheet omega glycoside that was utilized. These were both used in attachment to cage the dexamethasone, prednisolone, or the budesonide. Um, and then that is shown in D. You have the dex, the pred, and the bud there. Those are the drugs that were utilized. And the reason they selected these drugs is because, typically speaking, these drugs in patients who have suffered from IBD, IBS, or Crohn's have to take these drugs in lower dose amounts in higher dose amounts to get the same effect as they would in the lower dose amounts with the glycocaging. But taking them in such high doses means that the patients suffer from very, very severe side effects. And the side effects can be so bad that the patient wants to stop taking the medication, which means that their IBS, IBD, or Crohn's flares more. And so most of these drugs are only prescribed in like worst case scenario, really, really bad inflammation or episodes. But with glycocaging, the hope and the idea was that we would be able to reutilize these drugs and retest them so they could be more readily available for the patients, because these are some of the most effective drugs at treating IBD, IBS, and Crohn's disease. And then in E, we have the kinetic rates of the hydrolysis of dex, pred, and bud. And you can see the different Bacteroides species break it down at faster rates. So in this first one, the primary Bacteroides species they were looking at broke down the dex more, and then the secondary and tertiary ones broke it down a little less efficiently, but still efficiently enough to warrant the test of that specific drug with those specific Bacteroides species. So these are the results. This is figure 2. They did tests on 8-week-old RAG1 negative mice, which were injected intrapreneurally with the genes isolated from age and sex matched CD45.1 wild type mice to induce colitis. That is ulcerative colitis, which I showed you the image at the very beginning of the presentation. The mice were treated with the different varying solutions, micromoles of dexamone, or a vehicle control. So they used cyclodextrin as the control, or this 0.765 or 2.55 micromoles of triple X dex daily for 5 weeks, starting at week 4 after the T cell transfer. They checked for body weight changes in the mice. And those are the graphs in A. Those are body weight changes between the control, the ones taking regular dexamethasone, and the ones taking the glycocaged one. The glycocaged one is in the red. The orange is the straight dex. The green is the control, and the blue is the no transfer. So those are just normal mice. So, and then on the second graph, you see the same thing. Those that were taking nothing are in the green. Those taking the lower dose of the regular dex are in the yellow, and then the orange is the regular dex, and the orange is the triple X, the glycocage dex, and the yellow is the regular dex. And then in section B, we have the weight of the colon itself and the length ratio. So that is where the scientists, after the mice passed, would take out the colons, measure and weigh them, and that is in B. And you can see the triple X glycocage dex at the end has the lowest weight to size ratio, which is good. That shows improvement in the disorder. The dex ones, the purple, red, and yellow here that are hollow and not solid, are the dex, and you can see that they are slightly elevated in terms of the um, length to weight ratio. And then you have the control on the end, and you can see that the triple X dex looks more closely to the control, which showed that the glycocage dex did better at controlling the ulcerative colitis. And then in C, we have the representative hematoxylin and eosin stained cross section of the proximal colon for each group. So in the first one, you have the no transfer with the regular dex side by side. And that is the high amount of dex. So you can see that there's some good changes there. And then in the second one over here, you have the VC group. Then you have the high group, and on top is the regular dex, and on the bottom is the glycocaged. So on the top, you see there's still a lot of inflammation and a lot of issues going on. And then on the bottom with the glycocage, you can see that it looks more closer to the control. Less inflammation, less more gaps and striations, so that it's able to see that the drug effectiveness here was good, was effective in treating the disease before the mouse passed. On here we have same figure 2D here. We have the histological damage scores. So for the control group, obviously you don't have a lot of histological damage. Then you have the group that received nothing in the blue, and that has a very large score, obviously, because it received no medication but had the condition. Then you have the purple, red, and yellow hollow, which is the regular dex. You can see that it's obviously doing something and affecting it. But when you look at the glycocaged, you have almost back to control level inflammation and damage scores. So these guys have about the same amount of damage as a normal colon would have. And then you can see on the right of that, you have the same kind of cross section from C. That's from female mice, and the same thing that you see in the male mice, you see in the female mice. With the regular dex, there's still some of that inflammation. It's not as bad as the ones that are not treated at all, but it is very clearly not as good. And then with the bottom part where it says triple X G dex, you can see that it looks almost exactly like the control, which is showing that there is a profound effect on the fact that the drug has been glycocaged and is actually reaching the larger intestine. And then lastly, we have E at the bottom of the figure. The data are presented as a median interquartile range. So it's basically combining the two graphs previously and showing the control group in the gray on the left side of the graphs, the blue as the untreated colitis representative, the dex individuals that received the straight dex, and then the glycocage dex at the end. And the glycocage dex, you can see, is a very clear effect, whereas the dex has such a wide range of it depending on the dosage received. All right, so on to figure 3. With figure 3, we have the ship minus mice, which were treated with the same concentrations of the dex and the vehicle control, and then the triple XG dex or VC, which is water, daily for 2 weeks. A is the gross pathology of the distal ilia from one representative mouse from each group. So at the top, we have the vehicle. And then at the second line, we have the 0.76 dose. The third line, we have the 2.55 dose. And then the third line, we have the 7.64 dose. And essentially, you can see with the vehicle control, it's nice and healthy. There's not a lot of inflammation, and it's good to go. And then it's not good to go. It's very inflamed. My bad. I'm sorry. It's very inflamed and very not great. And then with the DEX, you still have some inflammation, but with the XXXG DEX, there's a lot less. The 2.55, you have a pretty decent amount, but toward the end it gets a little worse. But for the XXX DEX, it looks okay. And then for the 7.6, it looks pretty good up until the very end. And then the XXX DEX looks fairly normal throughout. The second part of this graphic, B, represents H&E stained cross-section of the distal ileum for one representative mouse from each group. The scale bars are 250 micrometers. So the vehicle shows the extremely infected mouse, and then you have the treated ones on the following sections. The top one being the regular DEX, the bottom one being the XXX glycocaged. And you can see that for the DEX, you have some effect because it's not glycocaged, so the medication is not getting a full effect in. But for the glycocaged DEX, you're seeing a much better effect and a much more relief of the symptom of the ulcerative colitis in the SHIP minus mice. Here we're going to look at C. The graph in C is the histological damage scores. So for here we have, again, the control, which is the untreated mouse. Then you have the DEX, and you can see there's a sweet spot in the DEX in the middle. The middle dose seems to be the best for treating it just normally. But then when you look at the glycocage DEX, the lower dose matches the middle dose of the DEX, which means that the lower dose here has been more effective at treating it. All of them have been effective. There's obviously lower than the DEX and the control, but with the lower dosage of glycocage DEX, you have the ability of the mouse to be a little bit more normal. And then in section D, you are looking at the IL-1 beta concentrations of full thickness ileal tissue homogeneous. So again, you have the controls in the blue. You have the DEX in the first section and the not solid graph bars, and then the glycocaged. And here again, you can see the lower dosage of the glycocage DEX is better than the others, but the XXX glycocage DEX is still as good, if not better, than the regular DEX, especially at the higher dosage. The higher dosage of the XXX DEX outscores the higher dosage of DEX by quite a bit. All right, and now we're moving on to figure 4. So for figure 4, they also wanted to take a look at the cardiovascular, or not the cardiovascular system, the vascular system, just to see what the lungs would look like on the specific drugs. One of the side effects is difficulties with air exchange. So SHIP deficient mice at 6 weeks old were treated with the drugs like previously stated. And then there was the vehicle control, which is the untreated mouse that carries the trait. So 4A is the gross anatomy of the lungs from representative healthy SHIP++ mice. These are mice that did not have the pathology. And then the DEX and glycocage DEX mice on the right. So here on the left of A, we have the healthy, non-diseased SHIP+. Then on the as we move to the right, we have the first control of the lungs. You can see the lungs themselves are also very inflamed. And then the dosage of DEX, you can see the lungs are still more inflamed than not. But then when you look at the glycocaged, the lungs look to be returning to a more healthy state. They're still inflamed, but they're not as bad as they could be. So that is also shown in the graph. So you can see the control score for the healthy individual is pretty low. And then the And then the DEX scores are pretty low because the drug is actually being delivered to the lungs and not to the large intestine. But with the glycocage, the DEX is still, the lung pathology is still pretty on par with the diseased, untreated individual. And then in B, we have the H&E stain sections from one representative lung lobe from one mouse per group. Scale bars are 1 millimeter. Percentage are H&E stain total and lung sections across treatment groups are reported as the mean plus or minus ST value. So here we have the vehicle. The SHIP plus again looks relatively normal and healthy. Not a lot of stuff going on there. But then we move on to the diseased lung of the untreated diseased lung of the mouse. You can see that there's a lot of swelling and the air pockets are not as dense. When we get to the regular DEX mice, we can see that they move more towards the vehicle, the unaffected vehicle, whereas the glycocage DEX, you can see that they're better off than the completely diseased one, but they're still not as good as they should be. And this is because, again, the medication is going where it needs to go and not going to be generally dispersed throughout the body. So with DEX, the lungs clear up a lot better, but with glycocage DEX, not so much. And now we're going to move on to figure 5. So with figure 5, they took fecal material from healthy control participants and people with IBD, and they were assayed for decaging activity using the triple XG res. There were no significant differences between groups as determined using a Mann-Whitney U test for raw data or an unpaired t-test for log transform data. So in the first image in A, you have groups that are looking at the resifurin and the supernatant and the pellet, and they seem to be fairly regular with each other. So they have an equal number of that in each group. In B, we're looking for calprotectin, and you can see again all the groups are pretty much the same across the board. It was, however, noted that IBD stratified according to fecal calprotectin. It's a marker of inflammation, so there were no significant differences between groups using a Kurzweil-Wallace test for raw data or a one-way ANOVA for log transform data. So the vertical lines are associated with histogram bars are standard errors of the mean on all of these. So the calciprotein was slightly elevated in the IBD individuals, but not a significant amount. So then we go to C. The presence of decaging endocyclokinase encoding genes for the Bacteroides species of control of ulcerative colitis, UC, or Crohn's disease individuals in the Human Microbiome Project, Part 2 study, determined by magic blast at 95% sequence identity and permissive 20% left and strict 70% right nucleotide coverage thresholds. So this is basically how they identified the bacterial species within the fecal samples they received from those individuals. Stratification of the GH5_4 endocyclokinase genes in individual metagenomes, that's in D here. That's how they tested and determined whether or not those genes were carried in those individuals. And then they did run a couple of short studies on the glycocage drugs against those fecal samples, and they did discover some interesting things, like it was effective in humans with those specific bacterial markers. So as for the discussion and a wrap-up, the results were largely positive and showed a decrease in the impact of side effects of the drug that were targeted and the overall health and well-being of those who were suffering from those disorders in the mouse models and human fecal tests. The results of the study show that that this therapy utilizes the microbiota in our gut to be more effective in assisting with medicine delivery. The study also showed that while the main target was for Crohn's IBD, there are other applications for glycocaging beyond the gut microbiome in terms of effects that would affect the rest of the body. So the drugs, when they were targeted to the large intestine, obviously, like we saw in figure 4, they decreased the effectiveness of that drug in other areas of the body, but the point of the drug is to target the IBD symptoms and the Crohn's symptoms, so it did end up doing what it was supposed to do. Conclusion: there is a glimmer of hope for those suffering from Crohn's and IBD and colorectal cancers thanks to this study. There's also hope for drugs that have severe side effects to be reconsidered for use in human treatments thanks to glycocaging and the human gut microbiota's ability to break down those plant polysaccharides. Here's a list of some questions for you to look over if you'd like to participate in a study or participate in a group discussion on these. What are other glycoconjugates we could look at in the future to assist with other absorption methods? This group targeted bacteroides. What other gut microbes could have been targeted? Why wouldn't bromide compounds be a good conjugate? The group focused on tamarin-derived glycoconjugate in this study. Why was it important to the success of the group to use tamarin? This group utilized a new method they dubbed glycocaging. Are there similar methods that are used now with other drugs in other places of the origin of the body to make medicine more readily absorbed? These are my references, and I hope you had a enjoyable time watching this presentation, and I hope you guys have a wonderful discussion.